It's really my very great pleasure to welcome you to today's inaugural lecture by the director of the Laufer Center for Physical and Quantitative Biology. Before we get into the main event, let me tell you a little bit about the people who made this event actually necessary. First are the Laufer family, whose generous gift made the center possible, and I ask you to please join me in congratulating them. Thank you. Thank you very much. I think we're very lucky to have this center, and the old saying goes that luck happens when opportunity meets preparation. And if the Laufers were kind enough to provide us with this opportunity, the preparation was set by two of our colleagues that I want to specifically acknowledge. The first is Carlos Leventhal. Carlos established computational chemistry as an important part of the Stony Brook culture and is himself a significant scientist in the field. And I'm fairly certain that we would not have Dill, our director, here today if it were not for Carlos' activities. The second person that I would like to thank at this time is Arthur Grollman. Arthur worked tirelessly, I almost said ferociously, vigorously to bring the Laufer Center to fruition. There are an enormous number of logistical and other barriers to overcome, and Arthur worked very, very vigorously to make this happen, and also lent an important scientific view to the center. So please join me in thanking those. And now for the main event, Ken Dill. Ken is considered one of the world's leading biophysicists. The citation that accompanied his recent election to the National Academy of Sciences reads, Dill is a pioneer in elucidating the physical principles of protein structure and folding. Through the insightful application of statistical mechanics, he shaped our understanding of how protein sequences encode their native structures, how proteins denature, and how they fold so quickly. The protein folding problem is considered a grand challenge in the field of biophysics. Scientists have long sought to understand how protein chains adopt the unique three-dimensional configuration that is so essential for their function. In 2008, the journal Science declared the long-standing protein folding problem to be essentially solved, citing Ken's primary role in the seminal research. Importantly, Dill's work has also led to the delineation of novel pathways, and it is an example of how basic science research founded in physics can generate new directions in biomedicine. Ken's initial contribution to research in protein folding drew upon his expertise in polymer physics, and that's appropriate as his scientific roots trace back to Bruno Zim and the Nobel Prize winner Paul Fleury, two pioneers in polymer science in the 20th century. After that training in the mid-80s, he designed an elegant model to explain the unique features of proteins which enabled them to adopt these well-ordered structures. And today, scientists from around the world in many fields use variants of Ken's model for studying protein and RNA folding, protein aggregation, and protein structure prediction. Ken's second important contribution also related to the grand challenge of protein folding, namely predicting native structures of proteins using only their amino acid sequences. Recently, using his expertise in physics, Ken and his group developed a computational algorithm that achieved the milestone of correctly predicting 8 out of 9 protein structures from their primary sequence. And the third major contribution, and then I'm going to stop because you can go on, but Ken's going to say something, was the creation of an entirely new novel field of research. He established the rules of folding that made it possible to design non-biological polymers that can also fold and mimic the functions of proteins and RNA molecules. These so-called foldimers are already showing promise in early trials as non-surfactant mimics, surgical glues for tissue engineering, and a new class of antimicrobials, all critical areas for clinical medicine. Not surprisingly, Ken has received many honors, including the first Hans Neurath Award from the Protein Society in 1998, which is one of the major awards in protein science. He received the 2007 Distinguished Service Award from the Biophysical Society for his leadership in our national science policy, and also, as I mentioned, was elected to the National Academy of Sciences in 2008. Finally, in addition to being a great scientist, I will also note that Ken is a great citizen of science. He has served in many leadership roles, including as president of the Biophysical Society, and he founded a coalition of basic research societies that promotes basic science funding and spearheads new federal legislation. Ken Dill has distinguished himself as a preeminent biophysicist and is widely recognized as a leader in the field of quantitative biology. We could not be more fortunate than to have him be our first director of the Laufer Center and to listen today to him describe his findings. Thank you.
Thank you, Eric, for that way overly generous introduction.
Fraction of the truth.
I, too, want to add my thanks to the Laufers, to Eric Kaler as Provost, who helped set up the Laufer Center, and to all the others that he thanked as well. So what I want to do is, I want to tell you, the story I want to tell you is called Pathways in Protein Folding in Evolution and Life. There's no way I would have been comfortable with such an incredibly presumptuous title, telling you stuff about pathways in life, if it hadn't been for the fact that we have a very broad audience, and it turns out that pathways in life happen to give me some great tools for introducing some of the terminology. So I apologize, that's why I'm doing it. So I want to tell you first about models. Models in physics and chemistry and biology. We all use these sort of kinetic models, and the basic idea is they describe states of systems written as A and B and C. In chemistry, these are molecular structures. In biology, they may be states of species or cells, for example, and they appear in physics as well. This is a common way of representing kinetic diagrams. And the pathway idea is the fact that you have something that is sometimes in chemistry, is sometimes called an energy landscape, and that's what's shown here. The energy landscape idea is that there is an energy, the up and down axis. Excuse me a second, I'm going to find a pointer. Is the idea that there's an up and down axis, and that axis is describing the favorability of a given state. Lower is better, more populated, so think of it, if you like, as water flowing along a little river or a little highway of some kind. And the basic idea of what pathways are all about is it's about starting at one state, one structure, chemical structure, and going through a sequence of chemical structures. And in chemistry, very often this is exactly what you find, are these relatively simple pathways that look like superhighways. Essentially, all the molecules start in the same place, all of them end in the same place, and they all follow the same route, like pouring water along a little riverbed. And it's that I want to focus on throughout the talk, the notion of pathways as these linear states. So when I started off as a little kid, I remember thinking that there was this thing that was called your destiny, and that when you grow up, you would have something like this, and this was here. This is where you start as a kid, and the issue is, how do you find this highway that gets you from wherever you are to your destiny? My biggest issue was I just didn't know where the on-ramp was. And what I discovered, though, over the years is it's not like what you think of at all when you're a kid, but rather, at least this is what mine looked like. What I think is this is going to look pretty familiar to everybody out there, so I don't think I'm particularly unique. But the pathways of life aren't at all like a straight line. They have all these other dead ends and that sort of thing. And so here's a little list of the things that I don't like about the linear idea of pathways, at least for life, and for some of the other scientific questions I'm going to raise in a minute. You have a large space of options. You make choices in your life. You make a choice on day one. Am I going to go to this school or that school? Am I going to major in this or that? Am I going to go to this city or that city? So forth. Often our choices are made locally, not globally, by which I mean you don't start by knowing where you're going to end up. I had no idea I would be at Stony Brook. I love it here, but I had no idea even a year ago I would be here, much less when I was 7. You make choices that are all local, that make sense at the time, rather than being driven by obvious global decisions. I'm going to use this idea of roll the tape again. This is something that appears in evolution all the time, and the basic idea is I want to distinguish two different kinds of things. On the one hand, there are what I would call micro routes, and that is something like my path through life or your path through life. That's a micro route. It's one trajectory. I'm going to distinguish that from a macro route. A macro route is something like, what would happen if I rolled the tape again? I started me at the beginning, and I ended me at the end, and then I did it again, and I did it again, and I did this in what a statistical mechanics guy would call an ensemble of times. So roll the tape again. your path would probably differ in detail. You make all these small decisions that end up leading you to a very different place. But if you roll the tape many times, there would be some universals that emerge. I suspect for many of you who are, most of you are scientists and engineers, biologists, chemists, and so forth, that what would have happened if you started your life over again, ended up where you are again, there would be some features the same. Maybe you'd still be in science, maybe you'd still be in engineering, things like that, but details would change. And the last point is that your end state, knowing the fact that you're at Stony Brook doing whatever you're doing, doesn't tell you anything at all about your route. And so this is, since this is a story about pathways, this is a story that's best told in road signs. I think this one says slow scientist crossing. I got this and some of the others that I'm going to show you from a great little book called Sign Spotting, so I want to give them credit. So I'm going to give you very briefly just an illustration of what I mean by these problems with pathways. Mine was this, very briefly. I was born a few blocks away from this bowling alley in Oklahoma, and if you're raised in Oklahoma, there's sort of two things you aspire to. Either you want to be a country western singer or you want to be a baseball star. So plan A for my life was I wanted to be Mickey Mantle, who, as you may know, was from Oklahoma. He was on the Yankees and he was great and so forth. That was my plan. But what happened is then I went to college and I thought my next plan was to be a mathematician. About sophomore year, I switched to mechanical engineering. By senior year, I switched to biomedical engineering. I then went off and did my PhD work. I was really interested in some rotations and origins of life research. My actual PhD was measuring DNA viscosities. Then I did postdoc work in polymer statistical mechanics, got to protein statistical mechanics, and at the end of the day, this was the story of my life. And I suspect it doesn't look too different for anybody else either. Now there's a couple of points particularly that I want to make here, and that is about convergence and divergence. So this is one of the situations you can have called a bifurcation. So even if we all started in the very same place, you could end up in very different places. All my buddies that I grew up with in Oklahoma are not in this room, for example. There's also convergence, and likewise all of those of you who are sitting in this room from Stony Brook came from very different places, I guess. And so, and probably not all from Oklahoma. I'm just taking a wild guess here. So this is sort of to introduce some of the terminology that I want now to get into something that's slightly more serious. And so first I want to tell you, I'm going to tell you 2 or 3 very small vignettes. And the first one has to do with protein folding, and that it is sort of the epitome, I think, of a story about these kinds of pathways and how they aren't always what you expect them to be. And in particular, the story, briefly, I want to tell you, since this is kind of older work now, I'm just going to summarize it briefly, is that protein folding has pathways also, and I will describe what I mean by that, but they're shaped like funnels. They're not shaped like superhighways from one point to another. And so the protein folding problem, which Eric mentioned, is the problem of taking an amino acid sequence of a protein. A protein is a chain of beads. All of the beads are amino acids. These are anywhere from, typically 50 to 1,000 amino acids long, and they fold up into 3-dimensional structures. You have about 20-odd thousand of them in your body, and each one folds up and functions in a different way. And the issue is, one aspect of the folding problem is, could we write a computer algorithm that could take this amino acid sequence, which is easy to get, and predict the 3-dimensional structure, which is much harder to get. It could be useful for things like drug discovery. At least that was the basic idea back when all of us got interested in this. Now, one of the other aspects of this problem is not so much the computer problem, but it is a kinetics problem. And that is, if I start with a chain and I pull it apart, which is the way it comes out when it's biologically synthesized, and I now wait some period of time, what happens is this thing folds up all by itself into this structure. And so there's the issue of the kinetics and the pathways. How does it do that? So there was this thing back around 1968 that started, which was the puzzle about, gee, how could this happen kinetically? It was called the Leventhal Paradox, and the paradox was based on the following idea, that if I think of a protein chain in a very coarse-grained way, so this is one amino acid here, another one here, and so forth, and these are all the bonds between them, and let's say I have 100 amino acids, then what I have is this huge space of options because it's like a piece of string that you can configure in a bazillion different ways. So if there's 3 options around each bond rotation here, which is ballpark correct, and I have 100 different bonds, then I would have 10 to the 48th different states. And so I have a haystack, and I'm looking for the needle, which is the one native structure, and the haystack is huge. And the question is, how would I find the needle in the haystack? So how does the protein, so the pathway issue here is, how does the protein meander through all of these various conformations and ultimately find that one right answer? What's really remarkable is this is called the protein folding problem, but it really should be called the people folding problem, because the protein can do this in of order 10 milliseconds. It has no problem at all. It's us that had the problem. So one of the ways of framing this so-called Leventhal paradox of, gee, how do I search this haystack and how do I find the needle and so on, was framed in terms of these energy landscapes. And now again, I'm showing you a diagram in which the up-down axis is a thing called energy. You want to find the lowest point. So think of this as a golf course, perfectly appropriate for the Laufers who love golf. You want to find the lowest point on this. So think of this, if you like, as a golf course, except that, and so the idea was framed in terms of a very flat landscape, but it's a 10,000 or 100,000 dimensional surface, depending on how much detail you represent the atoms and all the atomic motions. And so this is a landscape where you're looking to try to find the one hole. And not only is this area huge, but it's also very high dimensional. So this was the Leventhal paradox. How would you find a hole in a short time? So the key notion was maybe you would find this by the same narrative that I just described to you about life's pathways. Gee, maybe there's a sort of a destiny. Maybe I had a path, and maybe I could just, if I just found where the beginning of the path was, I'd be able to figure out the answer. And so the notion was, think about protein folding in the same way that you think about a chemical reaction. I start at one point, I end at another point, and that's because there's this sort of groove and the water all flows along that groove. So that was the notion that the solution to this Leventhal search paradox was to find a pathway. And so in the kinds of pictures I'm showing you, this sort of energy landscape, the idea would be, gee, maybe there's some kind of a route like this that flows down to the hole. That was one of the proposals that was popular at the time. And that was based on thinking about folding as if it were a lot like chemistry. The problem is that this is a very different sort of problem. This is not an A goes to B kind of problem. This is an everywhere to a certain state kind of problem. And so now I want to distinguish again this notion of microscopic routes, which is one road that one of us might take through our lives, for example, and macroscopic routes, which is some average over things. And so in this case, what I'm distinguishing is that I have one protein conformation will look like this, but there will be many, many others, and they will be points on that surface. And the question is, how do those other points get to the native structure? So this is not an A goes to native structure. This is not a one point to one point problem where a pathway would be the answer. Rather, this is an everywhere to somewhere question, in which case this isn't a solution to that problem, but there is a very simple one, and it's called a funnel-shaped energy landscape, and it looks like this. And you get this from doing statistical mechanics. We and other folks did this back in the 1980s, and you can see that proteins should have shapes like this. So now it's very simple to see why you don't have a Leventhal paradox, why it is that the protein doesn't actually take lifetimes of the universe to fold up, because think about taking a ball and rolling it around. You would never find the golf course hole here, because this is flat. On the other hand, you put the ball anywhere you want here, and it immediately goes to the bottom, and this just has to do with a little energy favoritism. Now, they're not quite so smooth and simple as that. They're a little bumpier, and this is sort of this is a picture that we looked at, and the gist of it is now that balls roll down the hills. Some of them get to this so-called native structure. That's the biological structure. Some of them get caught in these kinetic traps. But so the gist of thinking about the pathways now is it depends very much on where you start, what the microscopic route is that you take. But in the global sense, in a macroscopic sense, everybody can get to the same state, and they can do it very quickly. So what's the nature of the pathways? Well, proteins can fold along many of these microscopic pathways, not just one. And here's an example of what that sort of thing would look like. This is the native structure. This is a computer simulation by a German group, Frank Noe. That was published a year or two ago. A fair amount of computer time, because even for these short protein chains, it takes quite a while to simulate these under all atom atomic detail situations. But you can see the basic funneling idea. Here's the native structure. Here, many of the these are just some of the starting states. you can see that there's multiple roots. When you get down near the end, there's only a relatively small number of roots at the very end. And so it does start getting fairly systematic there. So this is a little movie that we made that shows you systematic, sort of a systematic random search along the energy landscape and showing you the corresponding protein structure and the pathway, the microscopic pathway it's taking to get to the end state. If you took even the very same starting point another time, because there's thermal energy and because things wiggle around, you would have a different route. So one of the problems in the folding field was that there are terminological issues, and they had to do with not distinguishing between these microscopic roots and these macroscopic roots. And so this is a typical kind of model that you would use in chemical reaction modeling. You would say denatured state goes to native state, and I have two of these states. And then sometimes, if you have interesting kinetics, you would have a third state, and you could draw a diagram in this way. This third state in this particular kind of diagram is called an off-pathway intermediate state because you really want to go from D to N, and what happens is some of these guys go off and they get stuck down here, and so they can't get to N without going back through D again. But this pathway terminology can be misleading. Like for me, for example, starting here and going to Boston for college and then San Diego for graduate school, what I would say is this was not an unproductive off-pathway intermediate for me. This was perfectly productive, and it was on-pathway for me. So the terminology that you get in thinking about these macroscopic roots and the microscopic roots can be misleading. So we've built some models in order to try to bridge between these two kinds of language. This is a case that shows you, this is exactly the same kind of model I just mentioned. What we're out to do in this problem is we want to find out there are proteins that are called ultra-fast folders. They fold in less than 10 microseconds. And for these particular proteins, it turns out they have this very unusual physical feature. They have this what's called anti-Arrhenius behavior. They don't have the normal temperature dependence that most all chemical kinetics would have. And the question is, how does that come about? And we made this model, and the basic idea of this kind of model is it's a way of trying to bridge between the macro and the micro. You can write down the differential equations in the usual macro sort of way, but when you interpret the rate constants, you interpret them in a microscopic way. This is just to illustrate that you can, in fact, capture this data. The anti-Arrhenius behavior is the fact that these curves, these black curves, have some curvature to them. They aren't simply straight. From the bottom left to the upper right, they usually have curvature. And this is a very atypical sort of physical behavior. And the main message from doing that work, and the reason that I'm showing it to you, is because this kind of mesoscale modeling, which can see both aspects, the pathways and the funnels, what we found is that if you use the normal sort of modeling approach, which is to think about things in terms of these single pathway Arrhenius kinetics, these things don't work very well. In fact, they actually blow up under certain circumstances, and so these turn out to be very bad models and require a large number of parameters. And that instead, the key thing that you need to think about here is this is about counting pathways. It's not about one route and trying to make a chemical model that captures one route, because it isn't one route. It's lots of microscopic routes. And instead, this just turns out to be sums over numbers of routes, and then you can model this very well. So let me just summarize this whole folding thing and just say that the folding pathways idea is that I have a search over a large conformational space of options. That's all the different ways this chain can be configured. There are many paths, not just one. In this particular case, there's many starting states and one end state, like all of us getting here at Stony Brook at the same time. Small bias leads molecules to a single native structure. You don't need to go from this flat landscape to a funnel-shaped landscape to go from infinite lifetimes of the universe time scales to 10 microseconds doesn't actually take very much bias. You just tilt it a little bit and the ball rolls down the hill, and that's all it takes. Roll the tape again. The individual folding path would differ in detail, but if you roll the tape many times, you get the experimental observables. It's the macroscopic kinds of things that you see in the experiments. All right, so now I want to switch gears to something I know absolutely nothing about, but I'm having fun with it, and that has to do with biological evolution and thinking about pathways in evolution as well. And here again, the puzzles are very similar. Is there a single pathway? Is there always a narrative in how one species goes from one, say, structure to another, or how cells evolve? can we rationalize the pathway by knowing what the end state is? And what happens if you roll the tape again? Would you get exactly the same route or not? And one of the key questions I want to raise here is these fitness landscapes. Maybe we instead of looking at them as linear pathways, this funnel idea that we can carry it over from protein folding, it does have a lot of the right features. So I want to go into an issue that's raised sometimes called the blind watchmaker argument about evolution. And the issue is, now I'm thinking about, this is a so-called fitness landscape, and so instead of down being good, it's up that's good. Up is fitness, and so the highest fitness is right here, and so this is called a fitness landscape. And so as species change and as mutations happen in your cells, what happens is you move along these things. Sometimes things get worse, which is going down, and sometimes things get better, and organisms get more fit as they compete against other organisms. So this is just a sort of a very schematic-y way of thinking about a fitness landscape. And the basic idea here is that an eyeball, how do we evolve an eyeball? Well, obviously it's a very positive thing for our fitness, and therefore this is probably at some kind of a peak, a fitness peak. But the question that's been raised often within typically sort of the non-evolution based world is, how would this ever have evolved? How would this have evolved through random stochastic processes that Darwin said evolution is supposed to happen by? And the reason that there's an issue is because if you made half an eye, it wouldn't do you any good. And so the question is, why would some organism spend all the energy and go to all the trouble to make half an eye if it isn't doing you any good? It's just a huge waste of your resources. So this is the issue of how do you rationalize where an eye comes from. So what I want to show you now is a little YouTube movie. And this movie was, I'm one of the talking heads in it. There's another one too. The other one is Eugenie Scott from what's called the NCSE, National Center for Science of Evolution in Oakland. She's one who watches out over intelligent design and creationists and so forth. But the argument that's made on this movie, it has to do with how it is that the eye might indeed have evolved. Let's see. Sorry, this is, hang on a second, let me just see if I can get an audio feed here. You guys have one? Okay, sorry. Sorry about that. Thanks. See complex structures like the eye having arisen from natural processes through small individual steps rather than from a single large step.
Mollusks show an interesting series of developments that probably also represent stages in the evolution of the vertebrate. First, you get a light-sensitive row of cells. And that has an advantage because that at least lets you tell light from dark, and that's important for some biochemical processes. Next, you have another mollusk that has basically a cupped structure where the cells are inside of a basin, and that allows you to tell which direction the light is coming from. That's an advantage over just being able to tell light from dark, so that could be selected for. Next along the line, you have basically the equivalent of a pinhole camera, where primitive focusing becomes possible. Next, you find the formation of a primitive lens, which again increases the ability to focus. And finally, in the last step, the octopus eye, you have a very complex structure with a very good lens, which is able to focus light very effectively on the back of the eye and thus transmit a lot more information to the brain.
Scientists not only see evidence of evolution in the increasing complexity of mollusk eyesight, they see it even in our own development. The eyes of a human fetus grow increasingly complex in stages, just as the eyes of mollusks do.
The first sequence was a schematic of what happens to an individual as that individual is growing in his mother's womb. First of all, you get basically a light-sensitive patch, and then you get a cup-like form that eventually forms a whole lens and a full eye with all the nerves and all the connections, bringing light to the brain and so forth. And then the baby is born, and then you can see.
What this shows is a complex structure like the eye can develop through many small steps from something simpler. And this is how Darwinian evolution works.
If it can grow, it can evolve. The same process, roughly, that produces an eye in an organism that's born, like you or me, is a very similar process that took place over millions of years across scores of different lineages of evolving organisms.
So one of the points that I want to make here is that in the business of thinking about biological pathways, there's often the sort of the battle of the narratives. One side has a narrative that says, here's how evolution happened. The other side says, no, here's how the process happened. And this is simply an example of a way of explaining a possible pathway. It's a linear pathway. And so now I'm also going to criticize what we just said in this movie. But a key thing that I want to leave you with here is there's almost invariably, if you look at an end state, like an eyeball, there's almost invariably plenty of explanations you can give for plausible pathways. And how do you prove one right or wrong? It's almost impossible to do it. It's just about plausibility. But I want to show you also, going beyond just the linear pathway idea now, something that is equally important, I think, and that is that these landscapes are big and complicated. There are many routes. There's a lot of options. And so there's another possibility, too, besides having just a straight line to make an eye. And that is that you repurpose and take a different pathway, and that this is really all about opportunism. This is about a process in which some organism makes some decision to fit within a niche, and the problem is there's no necessary narrative that's associated with that. Narratives are for us. They're not for evolution. Evolution may be doing things that are much more complicated. One of the things that we've been very interested in is bifurcation on fitness landscapes. This is a case where you have a choice of going to the left or right, and the decision you make is very marginal when you're making that decision. You don't really know what the height of the peak is when you go one way or the other. And these marginal decisions often have to do with noise and statistics and just flipping a coin and not having any sense of the final end state of the whole thing. There's a lot of very interesting non-equilibrium statistical mechanics in that, I think. And so the reason that I raise this now is simply because there are a lot of the features of these evolutionary processes that to us seem very similar to the folding problem. In the folding problem, we have these funnel-shaped landscapes where you're ultimately trying to get down here in the bottom. And in evolution, you have these fitness landscapes where you're ultimately trying to find your way to the top. But I think the shapes have very similar features in the sense that there's many, many, many states up here and few states here. And getting from the top to the bottom is a very similar process. This can happen very quickly. We think this can happen very quickly, too. We can use the same statistical mechanical models we've used over here. One is called the Zwanzig model, for example, for computing how long should it take to find the one native state in this astronomical sea of 10 to the 48th conformations. And it doesn't take very long. But I think the same thing happens over here, and for essentially the same reasons. If you have slight biases, even if you have sort of enormous numbers of possible options down at the bottom, slight biases can get you to the top very quickly. And so, for example, in a practical sense, you can see this sort of thing when you watch HIV virus evolve, or MRSA viruses, for example. These things can fill a niche in a very small number of replications. And so evolution isn't something that takes billions of years to go from one organism to another. Certainly, presumably for us, it took a while. On the other hand, if you look at bacteria, you look at MRSA, you look at HIV virus, and so forth, these things can become drug resistant very quickly. You hit them with a drug, you kill a lot of them, some of them aren't killed, and then what happens is the ones that aren't killed start evolving and forming new cells. And of course, cancer in the body is a very similar thing. You have some normal cell, it goes off, it wants to do something totally different, and it starts developing a stable niche. So what we think is this, in terms of these astronomical searches, this is something that, that statistical mechanics types like to think about a lot, are astronomically large numbers or infinitesimally small probabilities. And I think that this is going to be a case when we start getting better models for evolutionary processes, that what we're going to find is these incremental advantages can happen very quickly and can improve things very quickly. And along those lines, I just want to show you one calculation we did a few years ago. And this is, so let me explain what this is. We got some flack from the creationists, as they were called back in the 80s, for this, but here's the gist of what this is all about is the following question. The question is often framed, what is the probability that if I picked a random sequence of amino acids that it would look like a protein? And the answer that's usually given is this number is 1 over this number right here. You have 20 different amino acids. So in this string of pearls, each pearl can be one of 20 different colors. And if my string is 100 pearls long, then the total number of strings of pearls I have, or the total number of amino acid sequences I have, I've taken here a protein called lysozyme that has 130 amino acids. Then it turns out the number of different sequences is 10 to the 169th. So what's the probability of finding one of those sequences, the lysozyme sequence? If I reached into a big barrel and every sequence was represented in there once, we call that sequence space soup. The probability, of course, is 1 over 10 to the 169th. And so it's essentially 0. The chances of finding the lysozyme sequence are essentially 0. And so if you thought of space sort of as, gee, I have a sequence space, and if you think about space as being this huge dark sea, and only a teeny little speck every once in a while, that's kind of the view of what is the probability of finding an amino acid sequence that would look like a protein. But we asked a slightly different question, one that we thought was much more relevant in biology, and that is, I reach into this barrel, I'm going to still grab one sequence out of this large sequence space soup. But now I'm going to ask a slightly different question, and that is not what is the probability of finding the sequence of lysozyme, but what is the probability of finding any sequence that will fold to the lysozyme structure? Because biology doesn't care what sequence it uses. Biology cares what structure it gets, because that's what determines the function of the protein. Well, it turns out that that number is 120 orders of magnitude more favorable than the first number. So there's a huge difference. And the difference is very simple to explain. We did a lot of simulations. We compared them to a bunch of things. But the long and the short of it is it's really simple. And that is that, first of all, I don't need the 20-letter code. I don't need to pay attention to having exactly the first amino acid be exactly the right one and the second amino acid be the right one. All I need is roughly I have a 2-letter code instead of a 20-letter code. It just matters whether the amino acid is oil-like, called hydrophobic, or it's water-like, called polar. So you have what matters is, do I have a sequence that will give me the right hydrophobic core? Will put the right beads in the core of this protein? And the surface doesn't even matter very much. So once I go from a 20-letter code, 20 to the nth, as the size of a sequence space, to a 2-letter code, then you can see I reduce the problem by 10 to the n, and n in this case is 130. So I reduce the problem in a trivial way by 130 orders of magnitude, and so this is what we felt the sequence space actually looks like, is there's a chance of something closer to 10 to the minus 10 or 10 to the minus 15, you reach inside this barrel of 10 to the 169 sequences, that you would find something that actually could fold up to be relatively lysozyme-like. And what this indicated to us was that these fitness landscapes have, even though they have incredibly broad plateaus, those plateaus are actually sloping up, and therefore it's not so hard, after all, to find sequences that look very biological. They form structures, in simulations we found they form structures that would be called molten globules, which is, they're not perfectly like proteins, they don't totally lock in, but they're sort of wiggly and juicy a little bit, but from there, evolution could take them easily to a better state. So instead of thinking about this space of sequences, of amino acid sequences, as being totally dark except for occasional light specks, the way we see it instead is this whole space is sort of like a light beige, and there's some specks there that are even brighter white. And so evolution works on those kinds of sequences. So a lot of the work is not so hard after all. Let me show you the alternative. So one way to do this, one way to do evolution is by stochastic changes in these sort of random processes, ultimately improving incrementally as you go and finding good structures. That's the Darwinian approach. Let me show you what intelligent design gives us. All right, so now what I want to tell you about in the last couple of minutes is trying to do some modeling of evolution, and in particular, in this case, this is work of George Peterson, who's a graduate student in the lab. And what we're looking at is the protein-protein interaction networks in a cell. In this case, it's fruit fly. We looked at a few others, human and E. coli. And the question is, so what's known is, from proteomics and genomics, what's known is which proteins interact with which other proteins in the cell. What's not known is their evolutionary pathway, so we constructed a model to see if we could explore the evolutionary pathway. And let me just say a word about that, about these sort of diffusion processes in general, and then come back to that specific problem. That when you model one-dimensional diffusion, one way to do this is the so-called Smoluchowski equation. If you think about this as horses all starting at a gate, this is an x-axis, they're running along a track. So at time 0, they're all here. At a later time, they spread out a little bit, and they have some average forward velocity. And at some later time, they're even further down the road, and they're spread out even more. And so this is a diffusion equation with drift, it's called. So this is the diffusion equation part, and this is called the Smoluchowski equation. Basically, if you have data that looks like this, you have a diffusion coefficient, which tells you something about the width of this curve, and you have this average velocity, which tells you how the peak moves. This is the standard way we would model dynamical stochastic processes. One of the features, however, of this kind of model is that it always gives you these Gaussian distribution functions. And the part of the difficulty here is that many problems, particularly in biology and chemistry and in some of physics, and also in sociological problems, are not so simple. And they have these, instead of having these Gaussian distributions, they have these things called power law tails. This is a picture of a log of probability distribution as a function of stock price. So these are stock price variations over some period of time. And because this is a log scale, if this followed the Gaussian distribution, you would get this parabolic function shown right here. So if I used the Smoluchowski equation, which has been commonly done, if I know my stock price today, and I want to know what the errors on my estimate of the stock price is going to be at the end of the week, or the end of the month, or the end of the year, I could use this diffusion equation, and this has been a common way to do this. The problem with the diffusion equation is it gives you these Gaussian distributions, and so it estimates the width in a week or a month or a year based on how the Gaussian would spread. But if you look at the actual stock price fluctuations, this is this curve here, what you see is the data shows it's not Gaussian, and it shows these things called power law tails. And the gist of all of this, there's a number of books out there these days that have been written, and the gist of it is to show that we all tend to underestimate risk by tens of orders of magnitude when you're way out here in the wings. So things like stock market crashes, things like trying to estimate, when is there going to be a magnitude 9 tsunami earthquake combination, for example, hitting your nuclear power plants. We tend to underestimate by orders of magnitude the risk, and part of that is because a lot of what we do is based on these Gaussian functions. Instead, looking at the stock market, you see these power law tails. Also, if you look at the the protein-protein interaction connection networks. This is on a linear scale. This is a log-log scale. I don't have a comparable scale to this one, but this is showing you that these protein interaction networks have evolved in a way that's also more complex than a Gaussian would give you. In the last couple of slides, I just want to show you that we've been trying to model this. So far, we don't have a completed model of that. We have something that's closely related to it. And this is modeling the scientific citations. And when I say related to it, the reason that we picked trying to model scientific citations first is because there's huge data sets that are out there that are abundant, they're free. It's not like trying to get good stock market data. It's got this dual mechanism. It's got a power law tail. It's complicated in exactly the ways we want, except there's a lot of data and we can get to it. So we decided, okay, let's first see if we can model scientific citations first, and then we'll come back to the power law, to the protein networks. So this is work of, George Peterson also. You write these as difference equations. You end up getting this power law. Let me tell you what the essence of the message in it is that it's a two-mechanism model. And in this case, the two mechanisms are, when you write a paper, the first citations you get are somebody who knows about your paper or hears you give a talk. That's what we call the direct citation mechanism. After a while, when your paper has been around a while and when people get to know your paper a while, what happens is you get what we would call the indirect citation mechanism. And what that means is I find somebody else's paper who cites your paper, and that's how I know about your work, and that's how I cite your work. And it turns out the breakover point for when that starts to happen predominantly, when the citations start to come mostly from these sort of secondary routes, happens after about 20 or 30 citations. Let me just show you this. So this is the fit of the data, the model with the data. This is from all publications from the ISI index, showing you citation numbers. So there's some papers that are cited 1,000 times. There's very few of them. This is a log plot. Most papers get cited basically 0 or 1 time. And so this is also for chemists, highly cited chemists in 2007. This is for physicists and PhysRev-D over a 20-year period or so. Different data sets, but it turns out there's some universal features of these things. Some things depend a little bit on the field, but not very much. But the gist of it is these curves are all essentially identical to each other. This is just the integrated version of those curves. And the gist of what these curves show is a dual mechanism. And the dual mechanism breaks over right about where the knee of the curve is. So less than about 30 publications, people are finding your paper just by finding your paper. After about 30 citations, we call those classics. After about 30, what happens is now people are mostly finding your paper from everybody else. And many of you are well aware of this because you've seen some, they'll misspell your last name and then it gets misspelled in a bazillion other references that you see. And there's a number of interesting things about this, just from if you're interested in citations, which is not where we wanted to go with it, but it turns out that crossover point where things turn into classics is directly related to the average number of citations that papers make in general. And it turns out most papers cite about 20 or 30 other papers. If most papers cited about 100 other papers, then it would take about 100 for your paper to become a classic. So anyway, now everybody can go back and check your papers and find out how many are classics or not. So what we're doing is we're basically testing this kind of these pathways, these network pathways, on citations, and what George is doing now is to try to apply this to the protein nets. It's looking very promising, but I don't have good results to show you at the moment. The last point I want to make is that I think that these thoughts about pathways in general and considerations about pathways and funneling and so forth matters in other areas too. And there's this terminology called explore and exploit, and it's used in business and it's used in psychology, and it's basically that there's sort of two things you do. That if you're trying to find something, find an answer to something, let's say. First is you explore, and then you exploit. And explore means I have no idea what I'm looking for, and I'm going to spend some effort basically doing sort of random things. But then, and so I like to think of that in terms of funnels. The explore part is mostly when you're way up here on the top. It's pretty flat. You're looking around. You really don't quite know where you are. And the exploit is when you get down in here where you really can see the sense of direction, and what you're trying to do is just optimize, optimize, optimize in some way or another. And the reason that I raise this is because I think this comes up all the time as well in places like research funding. My personal feeling is that some of our funding agencies like NIH and NSF do lots and lots of support of this kind of thing, exploiting what you have, incremental change, trying to improve by one small amount from where you currently were, and too little of this up here, which is the explore, which is the real blue sky kind of thing. Now the difficulty, of course, is that the cost goes up exponentially up here. This is the z to the 48th kind of thing, and so the cost is proportional to that, so I don't see a simple solution to it. I just want to point out that I think these kinds of pathway issues appear a lot of places. And so finally, just to summarize then, my view about pathways is the case I've tried to make is that very often you have kinetic processes in which routes are through a large space of options. They're not always linear. They're not always simple narratives. There's often a lot of stochastic behavior in them. You can't predict the end from the beginning. You also can't predict the beginning from the end. They can bifurcate. Sometimes a small tweak can send you way off to the left or way off to the right, for example. And often there are local decisions that guide what's going on. They're not very guided by the global decisions. And finally, this is the gang. I want to thank all my lab group. And for the Kinetics Kings, and Huson and Banu were the ones who did most of the work I showed you. And then George Peterson and Steve Pressey for the work on the citations, and then for support. And I want to thank the Laufers and Stony Brook for having me here.